So good morning. Today we'll be doing our third lab, which is restriction enzyme digest. And for this lab, we'll be performing a restriction digest. We will use a restriction enzyme to cut DNA strands of two plasmids, producing a DNA fragment. Now, because we know the size of the plasmid and where the DNA will cut, we can predict the length of these fragments that will be produced. So for this laboratory, we'll be cloning the RFP gene, and this gene is derived from a sea anemone which is related to corals and jellyfish, and it reproduces a red fluorescent protein. So now we know the size of the pKNR, which is 5412 BP, and the fragment that will be cut is 807 BP, and the genes that are in between these is the RFP and the pBud. And the next one that we'll be using is the para, which the size is 4872 BP, and where the size of the fragment that will be cut is 377 dp. Now we move on into the materials, which is we'll be using buffers, pKNR plasmid solution, para plasmid solution, and restriction enzyme, also distilled water. We'll be using micropipettes as well. We'll be using empty tubes labeled already, A positive, A negative, K positive, and K negative. Also as other equipment such as the microcentrifuge, the trash, the floating tube rack, freezer, water bath, and ice buckets. Now we move on into the predictions of this. Since we'll be digesting using a BAM, HI, and Hindi on the two plasmids, which is the para and the pKNR, this is the structure that we'll look out. Based on the prediction we can make is that based on this plasmid map, how many fragments do you expect to create if both BAM, HI, and Hindi are used? Type your answer for each plasmid. So for the pKNR, we know that it is two fragments, which is the RFP and the pBud. Now for the para, we could see where they cut, and we see that there are no genes in between them, so we could predict that there will be no gene cuts. Based on this plasmid map, where are the expected fragment size that are produced? Now for the pKNR, the fragment that really produces is going to be 807, and we minus it from the total of the 5512, which gives us a total of 4705. Now for the para, we do the same thing. We minus the 277 from the 4872, which gives us a fragment of 4495 BP. What components will be found in each fragment? So for the pKNR, we could see that there will be the pBud and the RFP. pBud, RFP. For the para, where this cuts, we see that there are no genes, so no genes and no genes. Now we move on to the protocol. First off, we start off with selecting the water bath and placing it to a temperature of 37 degrees Celsius. Once that is set, we close here and move on to the next step, which is to select a P20 microliter, and we place the microliter to a 4 microliter setting. We save the volume, pick up the P20 tip box, and we get a tip box. And once that is selected, we move on to the next step, which is move the restriction buffer, here it is, to an empty rack. Alright, once that is set there, we go and we draw the 4 microliters from the buffer here. We push the button until it reaches the first stop. Slowly let go. And now we move the select the restriction buffer, and now we close this. After we close it, we move on to the next step, which is to select the K positive tube. Here we dispense the buffer that we just picked up, and now we push the button until it reaches the second stop. Once that this happens, we put the trash here and we eject the tip and we close the K positive. Now move on to the next step, which is to follow the micropipetting procedure to dispense 4 microliter of buffer into the K negative, then E positive and E negative tube. So what we do, we get another tip, close here, open the buffer, and we get more. Slowly let go, close the buffer, open the K negative, and let go. We dispense this tip, close the K negative. We open the tip box, get another tip, close the tip box. Get more buffers. Now this time into the A positive tube. Dispense the tip, close the lid, get another tip, close the tip, and get more buffer. Into the A negative tube now. So now we get that, dispense it. And now we move on to the next step, we eject the tip and we close the A negative tube. Now let's move on into the follow the micropipetting procedure to dispense 4 microliter of the PKNR plus mid K, which is this one here, into the K positive tube. 4 microliter, open the tip box and go back here, get more, first stop, we place it into the K positive tube. My bad, there's a here. Second stop, close the K positive and we dispense this. Now we do the same thing but for the K negative tube. Now once that is done, we check the tip and it says to dispense 4 microliter of the paraplasmid A into test tube A. Paraplasmid, which is this one, 4 microliters into the A positive tube. Go here, till which is the second stop, we let go, close this and throw away this, and we do the same thing for the plasmid A into the A negative tube. Press the first stop, we let go, close the tube and open A negative. We dispense this to reach the second stop. Then we place this in the trash, reject the tip and close here. Now we change the microliter back down to 2 microliter. We save the volume. Now we have changed it to microliter. Now we go to the restriction enzyme, which is RE, from the tube, and we place it in the K positive tube. I place this into the K positive tube. Now we just dispense the tip, close the K positive, and we do the same thing, but this time into the A positive tube. We get the restriction enzyme and place it into the A positive tube. When that is done, close both these and we eject the tip. Nice. Now we dispense 2 microliters of distilled water into the A negative tube. Get a tip, close it here, distilled tube. First stop, and we place this into the A negative tube. Once we do that, we distill this water, and we do the procedure again, and we place distilled water into the K negative tube. Once we do that, move on to the next step, but first we check the tip, and we close the one that we opened. Select this microcentrifuge to open it. We place this micropipette back into the wrap. We open it, we move the K positive, balance them correctly in placing tubes equal distance from each other. Now we go into the K negative, A positive, and A negative. Okay. So we put them in a way that they are balanced. I believe so, they are balanced. Check the balance, centrifuge look balanced and ready. So once that is done, we close here and we select it to close it. Select the microfuge to spin for 10 seconds. 10 seconds. Close it here and we start. Once this is complete, we open the centrifuge. I open it. We move the tubes into the floating solution. Tube wrap. Place it here. Okay, they are back. We close the centrifuge. Okay, so move them here. Select the microcentrifuge to close it, which I already did. Move on to the next. Select the water bath to open. Move the rack containing tube into 37 degrees. Now move it. Alright, in here, we select the water bath to close it. Set the timer by using the pulse to 1 hour. So we set this water bath timer to 1 hour. and we start it and close here. So when the time is up, remove the floating rock from the water bath by pressing the eject button. And so we just wait for the hour to pass. So once this passes, we eject it. We open the water bath and we eject. And I put this back here, close the water bath. So it says select the freezer to open it. Move the rock containing into the negative 20 degrees freezer. To open it and place it in there. Now we close it and next. This is our results. These are the actual results. Compare the actual results to the results of A positive. If their restriction was successful, you should see that the plasmid DNA has cut into the linear fragments. This is the ideal results and the predicted results. Now that we have completed restriction digest, which tubes will we combine to create new recombinant DNA? We could use K positive and A positive. Now move on to the next section. What do you think this is the main purpose of this simulation? The purpose is to learn how to digest plasmid using restriction enzymes. What is the purpose of heating the test tube to 37 degrees? This is the temperature at which restriction enzymes function at best. Submit the answer, correct. Why did we add the restriction enzyme to both pKNR and paraplasmids? This is to create the same sticky ends on fragments from both plasmids that can be ejected and joined together. Submit answer, correct. Why do you need to spin the tubes after adding your solution? This is to collect all of the samples at the bottom of the tube so that the reaction can occur, correct. What is the purpose of K and negative and A positive tube? This is to these tubes were used as a control to show the impact of the restriction enzymes and the size of the uncut plasmid. Submit answer, correct. Now for the feedback, I do feel confident, fairly confident that I could do this technique in an actual lab. Now what the next summary, which is a summary, and that was our lab for today.